Okay, hi everyone. I'm Rachel Hostetler. I'm from the Allen Institute for Brain Science, and I'll be going over some of the tools that the Allen Institute has to do some of the data sets that Michael mentioned is in his talk. And just a brief background about myself, I've been at the Institute for almost a year now. My role at the Institute is related to scientific training and outreach. So I function somewhat similar to an application scientist, where I provide training and scientific support to users of our tools and of our data sets. And yeah, and before that, I was doing my PhD work primarily in mouse, looking at characterizing different inhibitory interneurons. And I used a lot of Allen Institute data for my own PhD work. So I'm very excited that I can work here now and share these resources with others. And for those who are not familiar with the Allen Institute, it's a nonprofit independent research institute founded back in 2003. We're located in Seattle, Washington, and our structure is quite a bit different compared to typical academic labs. We have team science, which means we have very large teams, which do different portions of an experiment. So we have an entire team devoted to histology, we have an entire team devoted to transcriptomics, another team devoted to imaging, which is quite different from, a graduate student or postdoc doing all those steps yourself. We actually have devoted teams for all the different steps in our experiments. As you learned in Mike's talk, we also do a lot of big science projects, the very, very large scale projects that are worked on by hundreds of people. And we also believe in open science. So we're a nonprofit, all of our data is online freely for others to use and download. An example of that team science I'm talking about. So to do the patch-seq work, which again is a technique where you can get morphological, electrophysiological, and transcriptomic data all from one individual cell, there are many, many different teams involved with that process. We have a separate team specifically managing our transgenic animals. We have another team that works just on the tissue processing. We have our electrophysiology team, our sequencing team, histology, imaging, morphology. And then finally, to get all that data online for others to access and use, we have our data and technology team. So that's hundreds of people working on these data sets and on these projects. And here are some of the key people involved each step of these of this pipeline. And you were all introduced to the Allen Mouth Brain Reference Atlas in the previous talk, which is really our most popular tool. It's 20 years old. It features in situ hybridization data, and about slightly over 1,000 different mice were used to create this reference atlas. And even though it's 20 years old, it's still very much our most popular tool. But we have many more popular tools, newer tools with newer technologies. We have a lot of patch-seq data and a lot of different viewers to view that patch-seq data. We have our Microns Explorer, which features our electron microscopy data. We have some other types of data, specifically from two-photon imaging, looking at two-photon calcium imaging and electrophysiology in awake behaving mice, which is through our Allen Brain Observatory. And we also have our newest atlas, which is the Allen Brain Cell Atlas, or the ABC Atlas, which features spatial transcriptomic data. And we have more tools as well on our website. And again, these are free to use. You don't need to log in to use any of these tools. You can just access them freely through your browser. And here's one way to navigate it through our website. It's called brain-math.org, where you can find these resources. And this talk is specifically focused on cell types. You can click on our cell type logo here, which will then lead you to the different tools and data sets that are specifically for cell type focused. And we have a number of supportive resources as well. So we have a community forum, which is very, very heavily used by many of our users, where you can post any questions or comments that you have about any of the tools or data sets, and different members of the Institute will reply back to that community forum. We also have a new resource catalog, which is called our cell type supported resources, which is where we aggregated all of our different cell type guides and tutorials and webinars into one place to help people access and understand our data sets. And so now I'm going to briefly talk about how we define cell types at the Allen Institute. And we actually have a webinar on this on our YouTube channel. My team was involved with making a number of introductory webinars over the past year for those who are just learning about our data and resources. So if you want an hour-long webinar on this, we have that online. It's called Cell Types Taxonomy, A to Z Cell Types 101. And so at the Allen Institute, we really use single cell sequencing to define our cell types. And this is through the process of first, collecting the cells or nuclei from your sample, performing unsupervised clustering to see where your cells end up, and then organizing those clusters into some sort of hierarchy, and then annotating those clusters based on metadata, gene expression, and knowledge. And through that, we can then create these taxonomies based on transcriptomic data and see the hierarchical relationships between different cell types in the brain. And so again, we really anchor our definitions at the Institute with transcriptomic data. But transcriptomic data alone isn't, enough to know everything about a cell. We also need to know information about its anatomy, its morphology, its electrophysiology, its connectivity properties. So there are a number of other experiments and techniques that go into characterizing these cell types that are then anchored to that transcriptomic definition. So patch-seq is a technique that's an example of that. We have our cells defined by their transcriptomic profiles, but then we get more information to characterize those cell types based on their morphology and electrophysiology. And some central questions that we're trying to answer through all these different experiments is, first off, what is a cell type? That is changing, which cell types are defined in which ways, depending on which taxonomy you're looking at. These cell types have different names, they have different equivalents. How do we define and classify all these different cells? What do we call these different cells so that we all know what we're all talking about? How do these cell types vary across species and evolution? How are they generated during development? And then looking at the function of these cells. How do they work in circuits and networks? And what functions do they have? And also, the majority of our work is looking at, healthy non-diseased tissue. But we also have a few projects like our Alzheimer's project where we're looking at what happens to these cell types in specific disease states. And then finally, how do we selectively target these different cell types? What genetic tools do we have to target them, in a transgenic mouse or in, human tissue? How are we able to target and manipulate these cell types? And so now I'm going to introduce you all to the Allen Brain Cell or ABC Atlas, and we have another webinar on this on our YouTube as well. That's a full hour introduction into using this. And so what is the ABC Atlas? We all heard a little bit about it the previous talk, but it is a free interactive online viewer from the Allen Institute to view different taxonomy data sets that we have. And what's in it? As of today, there are 3 different data sets in there. When I first started working at the Institute a year ago, only one of these data sets was in the Atlas. And then so many projects have been completed in the past 12 months that we've actually added 2 more data sets to this Atlas in the last 12 months. We've added the whole human brain transcriptomic atlas from Sleddy et al. And we've also added our human Alzheimer's disease atlas. And then as more projects are completed at the Institute, we will also add those to the atlas as well. But right now, as of today, these are the 3 different taxonomy data sets that we have. And they feature slightly different types of data. So the whole mouse brain data features single cell RNA sequencing data and also MERFISH, which is multiplex error robust fluorescent in situ hybridization spatial transcriptomic data. And there are 2 main publications that summarize these data sets, Gao et al. 2023 and Zhang et al. 2023. These are part of a large Nature paper package last year. And then our whole human brain data, which features transcriptomic data as well. There's no spatial data in this data set. It's just single nucleus RNA sequencing. And that's from our paper Sleddy et al., which was part of a paper package in Science from last year. And then our human Alzheimer's disease, which Mike mentioned in his talk, is that CAD product that we had. It was accepted for publication pretty recently, but it's not in press yet. You can still view that paper on BioRec. And that features both single nucleus RNA sequencing data and that spatial transcriptomic merge data. And so first, I'm going to briefly describe the mouse data that's in the atlas. And so in this set of experiments, the single cell transcriptomic experiments were done first, and the data that's in the paper and also in this data set comes from over 300 different mice that were used for the single cell data, and 4 million cells passed quality control and were profiled. In addition, spatial transcriptomic experiments were also done, so another 4 million cells were profiled from a few mouse brands using a 500 gene panel to then integrate and integrate and map out the spatial locations of these transcriptomic cell types in the brain. And then by combining all this information, a hierarchical taxonomy was created, and there are a number of different levels to this taxonomy. So at the very bottom of the taxonomy, over 5,000 different clusters were identified. And then above that, we had supertype, which is another level. Then we have subclasses, classes, and neighborhoods. So there were 7 neighborhoods. Below that, 34 classes, over 300 subclasses, 1,000 supertypes, and then our 5,000 clusters at the bottom. And again, this was looking at the entire mouse brains. This wasn't just looking at one brain. We had a whole mouse brain was profiled for these experiments. And here's a demographic figure from that Yao et al. Paper that I mentioned that sort of shows the breakdown of the different levels of this taxonomy. So again, at the top, we have our 7 neighborhoods up here. You can see my mouse. Okay. So we have 7 neighborhoods up here, which are then split into our 3 classes, then split into our 3 subclasses, supertypes, and then clusters. And I'm repeating these because not all of the taxonomies that I'm going to talk about today have the same namings for their different levels. So this one goes class, subclass, supertype, cluster, but not all of them have that same name.
Hey, Rachel, what categories did you use to create those classes and subdivisions? When all the class and deciding this goes into this class and not this one, or that this is a class, not 2 subclasses?
Yeah, so there were a number of different people were involved with creating the clustering algorithms that went into determining these classes, and this is all available on our GitHub page as our package. It was an in-house created R package led by Vivian Yao, who's the first author on this paper, to look at what are the different cutoffs between those clustering. So it was all done. It's called Big Cat Scratch Taxonomy is the name of the GitHub, but I can also send that out to anyone has questions. But it was a evolution of a previous algorithm that was used for the clustering in the 2021 full cortex paper. This was something that was already in use at the Institute for a full cortex taxonomy, and then was expanded upon for these whole mouse brain.
Right.
Yeah. Yeah, of course. Yeah.
So, the fire starts from evidently known functional.
Yeah.
They exhibit, or leader, they exhibit obviously substantially different expression patterns. So there's a desire to approach it, but experience has taught us that to organize and make sense of data, it has to be done in some sort of hierarchical way, right? Now you look at neurons and you say, well, what do you have? And then you see the difference between the average and the loser. Well, when you continue in this way, of course, you keep going, and eventually you get down to some level where you have these base level kind of clusters. Now, that's the finest level that somehow statistically one is able to differentiate from them. Now at that level, still, I don't think we know to what extent how many of those are. The evidence that they seem to be distinct is one that statistically there are cells that group this way, and secondly, it's confirmed with other modalities such as epigenetic data that see similar movement. However, it doesn't mean that ultimately those would survive in some understood function. Now, as far as the intermediate level goes, there, you tend to see that if I take some of those lower level clusters and I group them together, my coherence increases. I've seen things that I can, and so this is just the way it's done, right? It's just this evident, kind of a somewhat way of doing it somewhat.
Yeah, and you can see up at the top are 7 different neighborhoods. These labels here reference, the names of different neighborhoods, right? So on the far right-hand side here, we have all our glutamatergic cells located in the palate. Then our next neighborhood over, number 2, is all our GABAergic cells located in the subthalamus. And then all the way on the left-hand side here, we have our non-normal cells, a lot of our microglia immune cells.
Well, when this follow up on genes, assuming all of this depends on which dimensions, features you choose to segregate. Right? So the question always arises, what if I chose other features, or what if I chose different weights for the features I have? You'll get a different clustering. So that's the general question is how fundamental are these clusters, or are they just a function of your choice of features and weights on those features?
Yeah, they're a function. They're a function of cell counts. Basically, a certain subgroup of genes are known to be the highest predicted in certain amount, say some 2,000-3,000 marker genes that evidently have strong differential patterns. And then using those, so you don't use the whole gene, we typically use about 5,000 genes, because those are the ones that through various statistical analysis are shown to be the most differential between different groups. And so then what you do is identifying that there are cells whose expression on the subset of the marker genes exhibits sufficient differentiation between another group that you would call indifferent. But it's all based on, at the level of the clinics thing, it's all based on kind of single expression counts. If you use epigenetic data, let's say a PAC-seq, there it's going to be based on alignment of peaks that are found in the kind of, in map genomic data to the genome. So it does very much depend on exactly what it is, completely data driven.
All right, I will move on to our next slide. So in this whole mouse spring data set, I mentioned that we have those clusters from our single cell transcriptomic data, but we also have spatial transcriptomic data as well. We actually have 2 different spatial data sets that are featured in the ABC Atlas. We have one from the Allen Institute from Lausanne, and then we have a second one from Xiaowei Zhuang at Harvard. And these are 2 different Merfish data sets, which I mentioned here because they feature a number of different genes and also a plane section and also a number of different donors. So currently in our atlas, we have 1 brain, spatial brain from the Allen Institute that's in the coronal section, and there are 500 genes that were used for that experiment. And then from Xiaowei Zhuang, that spatial data has over 1,000 genes, and there are 4 different mouse brains, and we have both the coronal and fascia. But they were integrated with the same single cell data set. So they have the same label of those different classes, subclasses, supertypes, and clusters. And if you're interested in learning more about, the finer details of how these data sets were created, the clustering, all that, here's a link down here at the bottom of the slide to that 10-page nature package, which features the Gao et al. Paper and the Zhang et al. Paper. And then also these papers also have links to those specific GitHub links to the R package that was talking about for the clustering as well. So a completely different data set that I'm going to be talking about next is the whole human brain taxonomy that we have from Slutty et al. which features single nucleus data from, as profiled across 105 different anatomical dissections in the human brain. And there were 3 whole human brains that went into this data set and 1 primary motor cortex from the 4th brain. And from these 4 different brains, over 3 million nuclei were collected. And this is primarily this was work from Sten Anderson's lab. And so there were different types of clustering that were done on this data set. And instead of having that subclass description that I mentioned before, here we actually have superclusters, clusters, and subclusters. So there are different names for the different levels of this taxonomy. Then at the end, we have over 3,000 subclusters at the lowest. And here we have in the Allen Brain Cell Atlas, this data set is actually split up between the neuronal data set and the non-neuronal data. And over, you could look up over 50,000 genes through this data set. And again, this was part of a major package that was published in Science last year. It was a 21 paper package. And here's a link below to access these papers. And then the third taxonomy that I'll be talking about today is our human Alzheimer's or CAD taxonomy. And this one is interesting because we can, by looking at this data set, we can actually see which cells were present in our CAD taxonomy that were not present in our reference data set. So we can actually see which cells are different in the Alzheimer's state. And from this data set, we can see that there were actually a number of microglia that were present from the Alzheimer's samples that were not present in the reference data. And also what's really unique about this data set is that 84 cells were collected, tissue was collected from 84 donors. And 84 donors spanned the severity of disease. So some had no dementia, no signs of disease, had low pathology. Others had very high levels of dementia. So we also have metadata about cognition of these donors before they passed away. So we had information on, their cognitive status, their number of years of education. And by having all this data, in addition to the transcriptomic data from these individuals, we can see where these Alzheimer's specific cell types appeared during the progression of disease. And as I mentioned, this paper was just accepted for publication in Nature Neuroscience. It's not in press yet. You can read about it on bioRxiv. Vito and Ciavaglini are the first authors on that paper. And in this data set, there ended up being 3 classes, 24 subclasses, and 139 supertypes. And this is specifically from looking at nuclei in the middle temporal gyrus and the dorsolateral prefrontal cortex. And in
Okay, oh, Alan, you want to read that microphone? So obviously they say that they just read the data, but the microglia, and did we not able to, did you have enough numbers to dig in and see if they correlate with some of the emerging microglia phenotypes that are coming up in models or generative models?
Yeah, I'm not aware of any comparisons to mouse models, but there was analysis done to compare this data set to, I believe, 10 other single cell data sets from human data from Alzheimer's to see if we look at the transcriptomic data, are we all recognizing the same microglia type that's different or appearing? So we actually do have that data that shows which ones are similar across these studies and which we're not, which is really exciting. So we can see, make sure, because we all give different names to different cell types. So if we say microglia 13, what does that mean?
Yeah.
Maybe it's microglia 22 in a different study. So yeah, that analysis was done looking at the human work.
Also, because the different subtypes are not only emerging as being anti-pathology versus pathology, but they're also emerging as therapeutic.
Yeah, and brought even more findings in neuronal. One of the results of the paper is identification of certain somatostatin by being early vulnerable, early kind of affected. Right? That is the result. We have found a novel study, but we also, in the study that she mentioned, it described a similar structure of the neural things like that. So we have integrated honesty into the existing study.
So one final question on this, not that I'm. With the way you do your sampling, I'm assuming you have frozen tissue or that you're taking information from. Do you have any way of going back? Those samples or samples from the same individual and correlating when you see some kinds of changes in microglial and correlating with loss of different neuronal phenotypes?
Well, in this first study, all this data was done in the middle. So it's all there. We don't necessarily have. We don't necessarily. In a molecular understanding. Exactly.
Okay.
Yeah, and with this specific Alzheimer's project, this is the initial areas that profiled for driver and dorcod prefrontal cortex, but there's a number of other areas that are currently right now being profiled and being analyzed in the lab. So this is the first paper on this study. It's not the end of it. There are many other areas that are being looked at in the hippocampus, visual cortex, other areas.
Are you doing any connectivity modeling with this? To look at changes in the
No. No. Not yet.
All right, and then just a brief description, again, that it's this data set that we have in the atlas. So we have both that single nucleus data and the spatial transcriptomic markers data, and we have that markers data specifically from a subset of those 84 donors that I mentioned initially. So of those 84 donors, 24, we also have the actual tissue samples, and they could look at the spatial location of those different cell types. So Mike mentioned that there are specific somatostatin interneurons that were noted to be especially vulnerable in Alzheimer's disease. Not all somatostatin neurons, just particular specific groups within this study. And you can actually see within the cortical layers of the tissue where they are. Because there was also a spatial component of, it wasn't just all somatostatin sprinkled throughout the layers. There were very specific layer differences between which cell types were more vulnerable. And that's what's exciting, having both the single nucleus data and the spatial markers data. You can actually see where those cell types are in the different cortical layers. And because we have the metadata and some information on these donors before they passed away, we have a lot of other metadata about them. You can sort through when you're using our online apps. They're all labeled by subject ID, so you can look at their APOE status. You can look at other pathological scores. You can look at, their Lewy body pathology, the age of death, sex, the progression score of the pathology, the years of education, the ROI that is used for the dissection. These are all different ways. This is all different information that we have on those 84 donors. And again, about 50% of them were cognitively normal, 50% had dementia, and then we had different levels of dementia as well. So we can really see which cell types are being affected at which stage in the progression of Alzheimer's disease. When do we see those new microglia appear? When are those somatostatin interneurons dying at the different stages of disease? Okay, so now on to the actual apps that I've been talking about. How do you use the ABC Atlas? We have a QR code here. I also have a link here. You could also just probably Google Allen Brain Cell Atlas and hopefully it will show up. Or you can go to brain-map.org, click on Atlases and Data, and then click on Allen Brain Cell Atlas. So first, before I actually go to the website, I'm just going to show a couple screenshots just to show what exactly is present, what type of information is on the atlas. So on our landing page here, we have a description of all of our different taxonomies. So the first one that I mentioned was that whole mouse brain taxonomy. Here is a paragraph here describing it. Links to specifically to data downloads on our GitHub is highlighted here. A cell type annotation table, which is extremely useful for learning more about the cell types, is available here. And then we have our different buttons to actually access the ABC Atlas. And once you actually click on the blue button and access the ABC Atlas, here's what it initially looks like. So we have, again, this is online in the browser, so you don't need to download anything. You can view both MERFish spatial data and single cell data side by side here. And right now, both these data sets are color coded by those 34 different subclasses. So the pink cells here are the same as the pink cells over here. And these are actually our our intertownscephalic and extratownscephalic glutamatergic neurons in this pink color. And you can see their actual spatial location here in the tissue section in the spatial transcriptomic data.
You should go on, you should go on and try that and play along with that. If you see, I'm sure you can follow more or less what she's doing.
Yeah, if anyone has any questions or they get lost on the website, we can stop and raise your hand. We have a lot of stuff on our website, having strictly defined things. Or yeah, just use this QR code, which hopefully you guys can. Oh no, that thing was blocking it. Okay, now you should see the QR code easily. And another thing is, it's helpful to have both that annotation table that I mentioned. We actually have links to that annotation table here. It was published as a supplemental table in the initial publication, but we think it's super useful. So we also put it online on our website. And we also made an acronym list as well, because not everyone uses the same acronyms to talk about different areas of the brain or different cell types. And so we created this acronym list to help better understand what these different acronyms mean. Okay, and then to actually use the atlas, you can color code things by these different ink drops here. So these ink drops just mean it'll color it based on what the category is. So I clicked on this ink drop here next to class, therefore it's colored in the data sets by the different classes. And if you were to click on this drop down arrow here, you'd see the other divisions of that taxonomy. So class, subclass, supertype, and clusters. And I'll actually go onto the website and demo this in a few minutes. I just want to give an overview first of what to expect when I actually get there. What else can you view? You can view the subclasses. So below our classes, again, we have like 300 subclasses. These are now different colors, and both of these plots are now colored based on those subclasses. Again, the same color mean the same subclass. So the blue cells here are the same cell types that are in our spatial data on the left. And here then on the left, you will see the names of the different classes. So for instance, class 1 is ITET GLUT, which stands for our intertownscephalic and extratownscephalic glutamatergic neurons. And this is why it's useful to have that acronym table open that I mentioned, in case you're not familiar with some of these different acronyms here. You can also color the plots, these cell types, by the neurotransmitters that they express. So before, I was just coloring everything based on what cell type they were based on transcriptomic data. But let's say you just want to look at the gathering cells, glutamatergic cells, or those that express dopamine. Here now these plots are colored by different neurotransmitters. And glutamatergic here is represented in blue, GABAergic is red. Cells that don't express neurotransmitters, those are colored here in gray. You can also color plots by genes. So again, we have over 30,000 different genes that are present in these data sets. We can actually search different genes of interest. So here I type in CALB1 for calbindin. And you can see where in these cell types, which cell types express this gene. And the way that it's colored here, the darker color means higher expression of the gene. And in our single cell data, we have, again, over 30,000 different genes. But the way that spatial transcriptomic technology works, you cannot, currently, there's no technology to profile all 30,000 genes using spatial transcriptomics. So we're limited to our 500 genes. So the data set there is slightly different. We have a number of scientists currently working at the Institute to get our imputed genes linked with these spatial transcriptomic data sets that you can actually view all 30,000 in it, but it's currently not live. It will be very, very, very soon. But right now you can just look at the 500 genes that are profiled in the spatial data set. And I mentioned earlier that there were 2 different spatial data sets in this atlas. There's one from the Allen Institute, which is from Humphrey Zhang, and then there's another one from Shuawei Zhuang from Harvard. And in Shuawei Zhuang's data set, there's actually 1,000 genes that are profiled in their spatial transcriptomic data set. So sometimes the gene is not present in the data set you're looking for, it might be present in the other data sets. You can also color plots by the different dissection regions. So the way that the spatial experiments were done, it was mapped with that mouse reference atlas CCF that Mike mentioned in his talk here. So that's color coded by the different anatomical regions. But then our single cell data was profiled based on different microdissections of over 300 different mouse brains. And so they are dissected in slightly different ways and labeled slightly differently compared to our spatial data. And this is all color coded here, so this is by anatomical division. And if you have our mouse reference atlas open while you're looking through this, this will be really helpful to understand some of these acronyms. A few weeks ago, someone was using this and they thought TH down here meant tyrosine hydroxylase. It does not, it actually means thalamus in this case. That is one example of why it's useful to have the reference atlas open when you're looking through all of these different acronyms. So some of these acronyms might not be used by everyone. You can also isolate different areas. So if you just care about primary motor cortex, you don't care about other regions, you can click on this little box here, check box it, and just isolate cells from that anatomical area. And then once you have that isolated area, you can then also color code your genes as well by looking at, I only care about calpain in primary cortex. You can also duplicate plots. So right now I have two different view maps featuring that single cell data duplicated next to each other. So here you can look at and compare different features. You can have one plot showing, the different classes. You can have another plot showing gene expression for a particular gene of interest. And then you can see where that gene of interest is that expressed in multiple classes or subclasses, or is that restricted to a certain area? Is that a marker gene that would be useful for your experiments? And you can also color plots by different genes. So here I have calpain and calretinin side by side, because the question you might have is, are calpain and calretinin co-expressed sometimes in certain cell types? That's a question you could answer by looking through this data set. Some other use cases. So again, are by you can have four different windows open at the same time. So you can look at four different genes and say, are these four different genes in the same cell type? Where is gene X in the brain? Is gene X in a specific cell type? Where are these cell types in the brain? Are they just in specific cortical areas? What's their spatial relationship? Are they within the same cortical? Are they next to each other? These are types of questions that you can answer using the atlas. There are other questions that you cannot answer. So the type of data that we're showing here is really looking at, based on cell segmentation of the cell body. So you're not looking at axon terminals, you're not looking at dendrites. And so if you're really interested in synaptic information, you cannot get, we don't have that resolution here with spatial transcriptomic technology to try to answer those questions. And again, when you zoom in and out on this data set, you're not actually looking at the size of the cells, because those cells sizes will change as you zoom in and out, because what's shown is the centroid of the cells. So if you're interested in the morphology of the somas of the cells, you will not get that type of information from the cells. Okay, so now I'll actually go and show what's available here. So again, here's that landing page. Here's a little YouTube tutorial as well. And again, our different data sets. So here's that whole mouse brain data set that I mentioned. Here's that second Murfish data set from Charles Wong from Harvard. Here's all the Alzheimer's data down here. And then our transcriptomic from our adult human brain from Sleddy et al. Is down here. So everything's laid out first on this landing page. And we have a few quick start guides as well to help introduce people to the data sets. So I'm clicking on mouse because that's what I'm most familiar with, but I don't know if that's useful for this group. So someone wants to raise their hand, or like, who would be more interested in the human data, the Alzheimer's data versus mouse data? Like, anyone can shout out about what would actually be useful. I don't have to go through mouse data if that's not actually useful for people. People interested in the Alzheimer's data, perhaps?
Yeah.
Okay, I heard one yes. That's enough for me. I will. How do I get back to my screen? Okay.
Well, the good thing about looking at that, we can, so the Alzheimer's Center, remember what I said, the Alzheimer's data, the transcriptomic part is all collected in middle temperature, the first region that we profiled. And it was done by, we had the structure, of the normal. And we then did all the individual donors. We found that we could extend certain, to James' point, extend some of the wheel type off in this donor data. But the thing of looking at this is it provides, you get a sense of looking at the omics data from the perspective of the default atlas or the normal typical. And you also get to look at the methodology and then something. We can probably do. You need to review some of that.
Yeah, so here on the left here is our, is the spatial transcriptomic data looking at tissue sections from, yes, the middle temporal gyrus here. And then on the other side here, we have our single nucleus data from both the middle temporal gyrus and the dorsolateral prefrontal cortex. And by going through and filtering, so if we're interested in looking at those particular microglia, we can actually go through here and look at, right now this is colored by different subclasses of cells, but I can go and click class. So the three classes that were divided up, that were established through this analysis were our neuronal cells that are GABA, our neuronal cells that were glutamatergic, and then our non-normal and non-red cells.
So we probably need to build it. Yeah. Because I only have access to the single cell data.
Oh, okay, did you click on it and only one window popped up?
Yeah, I think so.
So if you go up here to this symbol.
Yeah.
Our view manager, you can actually click add visualization, and if you search through here, you should be able to click, yeah, this is showing all the data sets, it's also showing all the mouse work. But all the way at the bottom here, if you click CAD, MERFISH, MTG. And again, you can have up, oh, that's not good. What was that? You can have up to 4 different views on here, so I need to go back to the. Almost never see that. Okay. Yeah, you can also exit out of this. You don't want to look at both the single cell and spatial data at the same time. You don't have to, but being able to add different visualizations, you can do that through here. And also change which way they're showing too, like a vertical or horizontal. We also have night mode, which some people get really excited about whenever I point that out. Okay, yeah, there's some vigorous head shakiness. So that also changes things. You want to use the night mode. But again, going back to those specific microglia that we're interested in, if we go down here and we zoom in on our sample. So here, these are actually tissue samples, and these are from our 24 different donors. And you can actually go down here and click on subject ID. And if you click the ink drop, it is now coloring those different donors, coloring the tissue samples based on the different donors. So any, these 2 light green, these are from one donor. These 3, yellow green are from one donor. These 2 red ones are from another donor. So here you're looking at all 24 donors at the same time. And then in this single nucleus data, we have 84 donors here, and these 24 spatial are a subset of this 84.
Rachel, what question? These are all Alzheimer's or some of these age?
So all of these, yeah, not all of them were diagnosed with dementia. About 50% of them were actually considered cognitively normal.
Okay.
But then you can also, you can do that here actually by looking at, let's see, cognitive status, right? You can color that as well. You click on that ink drop. Now we have dementia versus non-dementia. So this first person here, dementia.
So do you have a way of going in there and looking at some of these subtypes? Either neuronal or non-neuronal?
Yeah.
To the other one, your data sets?
Yes. So I'm going to duplicate this here. I'm gonna get rid of this this UMAP over here just for a second. So I duplicated it here because I think that's a very useful question. Look at things side by side. So right now this plot here is colored by those with dementia, samples with dementia versus no dementia. And then if we go back over here, I can now color things by the different microglia that we're interested in. So right now, in our non-neuronal group, we have, here, I'm just gonna zoom in on. Oh, actually, I can actually sync these two, so it's easier to see, no, what's going on here. Camera. Okay, so now I'm gonna zoom in. Because I'm really interested in like these two different sections. These are from two different donors. One had dementia, one didn't have dementia. You can go in and look at just the microglia. If I go down here. This is our microglia PBM group here. And I can get rid of all the other cell types. So if I'm not interested in astrocytes, endothelial cells, right now I have just my microglia that are shown here. And the ones that say CAD, those are the ones that were specifically identified in the Alzheimer's group. So microglia, micro PBM 2-1 CAD, this super type here was found to be an Alzheimer's specific microglia group. Same with down here, microglia 2-3 CAD. So if I want to just click on one of those, I can exit out of the other ones. So right now I'm just interested in this group here. And I can actually zoom in. And you can see all the other cells are sort of grayed out here because they are not selected in that group. But the ones that are there are part of that CAD group. And then I can highlight them. You see when I hover the cursor, the dots get larger, the cells get larger. And then I can look at my panel next door and see where those cells were. Maybe there were more, where were they located spatially in the different board layers. So that's one example you could do.
So in your paper, did you do any cross analysis across individuals that are critical, that kind of, I mean, I don't be able to tell you, you could do brain imaging and 3 individuals who seem to have the same symptoms of Alzheimer's or dementia, and it's really hard to correlate across. Why? Even with, high-end MRI, and have you looked to see test the individual variability in any of these cell types across the individuals who seem to be staged similarly? Or is that left for the rest of us to do?
Well,
Yeah, you probably better answer.
I mean, there's all the metadata is all there, the patient data that you can map against, that you can track. It's not, we don't have to be very interested. We don't have a deep analysis of neuropathology, corresponds to that. The action they actually to actually do would be to actually go through and using some, machine learning techniques. And we extract all imaging data, which is all available on in an enzyme, and focus on the responses. So that can be done.
Right.
Yeah, because while we're on this topic, I do want to mention that this is not the only data available from this project. This is just the single nucleus and spatial transcriptomic data. But if you go to CSEAC-AD.org, you will get to our CAD landing page, which is what I'm showing here. And we do have some neuropathology data here that you can actually view. If you click on our resources here, this data is not only present in the ABC Atlas, it's present in a number of other viewers. So here's another way to view the transcriptomic data. All the way down here, we also have it on cell by gene. Down here, you can actually access the neuropathology viewer. We have information on the donor index. And also epigenetic data as well. So that's not featured in the atlas that I was just showing, but this is other data that was collected in these experiments that you can access through other ways as well. So if you're interested in the ataxic data, I do have that. If you're interested in some of the neuropathology data, if you click on the neuropathology image viewer here, you can actually view different sections, different stains that were done. You can look at the plaques, and again, we have the subject ID for all of these individuals. So H2133045. You can find that same donor and look at the transcriptomic data in the ABC atlas, and then look at, the actual neuropathology data here as well. So they are all linked across the different ways. But yeah, but this isn't in the ABC atlas, this is in a separate viewer that we have.
All that division, so that in the neuropathology data, there's that was done in 13s, right? And they use all this HIBALL suite, which is kind of a quantitative suite that you can actually, count various kind of stains for, plaques.
Parts of there.
Right, exactly, like that. And so that, all that data is actually available and numerically also, so there's a lot to be done with that.
Yes, and if, you're interested in downloading like the transcriptomic data from these 84 individuals, that's also online. It's hosted online through Sage Bio Networks. And so for that, you actually do have to register and just fill out some information, since that's personal information from these individuals. That's another way to access that data, and that is through, Sage Bio Networks. And to download any of this, if you have any questions on how to access things, this giant logo here is showing how to get to the data or download. And if you click down here, you click explore, this actually shows you how to access all the different data types. So if you don't want to use these online viewers or portals, you have your own analysis, we have links to all of that as well. And also the white papers. So the information, these are very detailed protocols and methods of how the analysis was done, how, the nuclei were extracted, how things were analyzed. So we have all of that located down here as well. More detailed information about the cohort, more information about the metadata, more clinical information, all that is on this website. Yeah, so there's a lot of extensive data from this cohort that isn't in the atlas that I'm featuring, because this atlas is really just designed currently for transcriptomic data, but we have all this other data online for others to really access as well. Okay, so I'm going to go back to the presentation. So that's the Allen Brain Cell Atlas. If there are any other questions about that, then I'll be here for the rest of the workshop. So you can also talk to me today or tomorrow or Wednesday. But I'm going to move on to one of the other tools that I'm going to be demonstrating. Oh, we also have tutorial notebooks. If you're familiar with Python, we actually have a number of notebooks where you can just access all the data without needing to use that browser, use that portal that we created. If you want to do your own analysis, we have some notebooks to help out with that as well. And that's located on our GitHub, and here's also a link to that. And these notebooks, right now, the screenshot is just showing that mouse brain data, but we also have notebooks for the CUNY data as well. And what's next? So again, that's what we have on the Allen Brain Cell Atlas right now, but there's a number of other directions that we'll be going in. So another direction is perhaps integrating generative AI to make this portal more accessible. That's something that our data and technology team is currently exploring, trying to make things more connected with other data sets, adding in more data. So Mike mentioned all that basal ganglia data that's currently being analyzed at the Institute. Once that is done and we have a taxonomy for that, more data sets will be added to the ABC app. So as more data comes out, we will keep adding other data sets to it and try to make it, more user-friendly, more interactive, so that others can use it for their own research. And also more developmental timelines. There's a mouse developmental atlas that will be added sometime in the future, to this portal as well. And this is just a slide summarizing some of those CAD resources that I had just mentioned. So again, c-ad.org. This is where you can access some of those plots, some of those different data sets. And down here, actually, in this bottom corner, right here, we have this is actually a progression plot which shows these different cell types, how they're affected throughout the course of disease. It's very small here because I just took a screenshot to highlight it, but this is where you can actually see which, glial cells are increasing throughout the course of disease, and which interneurons are dying, which are more vulnerable. So you can look at all that in this progression through our different plots that are on c-ad.org. And that was combined, that was analyzed by looking at the transcriptomic data in addition to the neural pathology data, and seeing where do we have that association between cell types and the disease state of the brain. Okay, next tool that I'm going to talk about is Map My Cells, which you briefly heard a little bit about this morning. So Map My Cells is a tool where if you have single cell or single nucleus transcriptomic data, you can map it to the taxonomies that I just mentioned. So that mouse taxonomy, the human taxonomy, and the Alzheimer's human taxonomy. Those are the three different transcriptomic taxonomies we currently have on Map My Cells. If you have your own data, you can map it to those taxonomies, which is useful, if you're studying, microglia and you're saying, oh, I have microglia 12, I'm interested in how it compares to the Alzheimer's data, you can map it and see, does that compare to the Allen Institute Alzheimer's data? Is it the same type of microglia? Is it not? So the whole purpose, again, is to label, if you have unlabeled cell by gene data and you want to annotate it with the cell type names from our taxonomies, this purpose of using Map My Cells. Map My Cells was actually used during the creation of this whole mouse brain atlas to map that single cell and spatial data together. So again, we have those three taxonomies that I just mentioned that are featured in Map My Cells. And how do you use it? So this is also browser-based. You don't need to download anything for this. You just go to brain-map.org, click on Atlas and Data, click on Map My Cells. This was designed to be very user-friendly, so you just need to drag and drop your file into this little box here. Click which taxonomy you'd like to map to, click which algorithm, and then click Start. And it takes about 5 minutes or so to do the mapping. And there are some input file requirements. You can't just drop any type of file into the upload link. It actually has to be a specific file format, which I'll go into in a little bit. And if your mapping doesn't work for some reason, what happens is you'll get this little box here that says mapping failed, and it'll give you this code. And this code is actually super useful because I mentioned we have this community forum that people use a lot. You can go to our community forum and say, hey, I did this mapping, here's my code, what went wrong? And one of our scientists involved with Map My Cells can then look it up, see what went wrong, and try to troubleshoot for you. And the other information is not saved. Whatever data you upload in here is not being saved by the Allen Institute for any reason. It's briefly, just that code is saved for a little bit of time after you do your mapping, just in case something goes wrong, but we're not permanently holding on to anyone's data. And if you're concerned about that, we have this statement here that goes into more detail about the data rights. Then what's the output? So after you use Map My Cells, if it maps correctly, what you end up with is a CSV file and a JSON containing some metadata. And the CSV file shows a number of different things. It shows, here this would be your cells that in the data that you uploaded, it has to be a cell by gene matrix. So your cells would be labeled here on the left. Then you have your machine readable assignments here, which these orange arrows are pointing to. And then you have the human readable assignments, which are in red, so the actual subclass, class, supertype names. So the way that this mapping works is that it doesn't just map and say, cluster X is equal to cluster 13. It actually says cluster X is in class A, subclass Z, supertype X. And finally, cluster so and so. So it goes through each level of the taxonomy and will map your data through that. And then you also get a confidence level in your mapping for each level of that taxonomy, which is what's shown here, what the blue arrows are pointing to. And so what is it doing? So there's a number of different algorithms that were created by our data and technology team for this mapping, depending on the taxonomy that it's being mapped to. So we have our deep generative mapping, we have our hierarchical, and our flat mapping. And I'll briefly just talk a little bit about hierarchical mapping and how the hierarchical mapping and how it's working in this algorithm. And also, this is also available on our GitHub. So another thing is, if you're using the browser-based system that we have, there is a 2 GB limit. But if you're good, with coding yourself, you can actually just get the code from our GitHub And then you're not restricted by that 2 GB limit anymore. And so for how the hierarchical mapping works is that first it's looking for different marker genes. And it's looking at different genes, marker genes, of how to discriminate these cell classes below the current node. So right now, in this example, we're looking at, if I have a cerebellar neuron, what type of cerebellar neuron is it? And so it'll look up different marker genes that are associated with the classes below cerebellar neuron. And then it'll do this mapping 100, the algorithm will then do this mapping 100 different times. And you'll get different confidence values at the end, looking at the lowest node on the taxonomy here. And after it's done this mapping 100 times, we can see that, 81, we have the highest correlation here with granule cell cluster 2, which means it's going towards this subclass. And then this mapping repeats 100 times at each level, which then finally gets it down to the individual class. So again, your data is not just mapped to, the cluster at the very bottom, it's actually mapped to all the different levels of the taxonomy here. And this is where that confidence score comes from. You can see out of 100 times at each level, how many times did it map to that particular level? And Scott Daniel is one of our software engineers who was involved in creating this graphical mapping and creating this algorithm here. So he's also very active on our community forum. So if you have any questions on your mapping or questions about the algorithm, you can definitely reach out to him on our community forum and just ask pretty much any question about Map myself. Or you can read through other questions that other people have asked about Map myself. So that's another good thing about our community forum is that you can search through about 5 years of questions that people have asked on this forum about different things. Map myself is not 5 years old, it's under a year old, but if you have questions about any Allen Institute things, I highly recommend searching through our forum to find answers. And another question is, how well do these mapping algorithms work? Right, that's the number 1 question. How can I trust this? Why should I use it? And so a test that Scott actually did was to take a subset of that whole mouse brain taxonomy data, take a subset of those cells, and then map it back to the whole taxonomy and see how well does that work. And another question is, how many marker genes am I supposed to? So in the whole mouse brain taxonomy, over 6,000 marker genes were used to discriminate between the different clusters and the different levels. And perhaps you don't have 6,000 marker genes in your data set. What happens if you only have 5,000, 4,000, 3,000, right? So Scott actually went through and mapped out these different levels. And even if you only have 3,000 marker genes here on this graph, you can still see it actually still maps pretty well, even though the algorithm expects to have the full 6,000 genes. Other quality metrics, how exactly does this mapping algorithm work? So at each point, it looks at the marker genes associated with a certain cell type at a certain node in the taxonomy. Then it selects randomly 90% of those genes, then it maps 100 times. Depending on that, we'll move down to the next node. And then it keeps repeating this and repeating this, and that's where you get that probability in your confidence level. And more questions on, more links about how to use this. To access the online app here, our community forum, the actual code, and documentation on how this works. And I have talked about Map myself in a few different conferences, and I have found people are using it in a number of different ways. Some people are using it, trying to use it for cross-species comparisons, since we have both mouse and human taxonomies to map to. I talked to another person who was creating their own developmental transcriptomic atlas of the mouse, and she was using this code to map her cache-seq data onto her taxonomy data. So people are really using this code in different and unique ways. And you can do that if you're not limited by the browser. You can just download the code and map it yourself in any of these ways. And again, the input file, there are some limits. It needs to be in an H580 file format. It needs to be a cell by gene matrix. That's what it's expecting. It also needs to be single nucleus or single cell data. If you use bulk, transcriptomic data, it will not work. It is expecting individual cells. And again, the output is a CSV file where you'll get your cell IDs labeled with the Allen Institute labels, and you'll get a JSON file as well. And once more taxonomies are created, just like the ABC Atlas, they will also be added to MapMyCells. So when MapMyCells first came out, initially did not have the Alzheimer's taxonomy in it, nor the human brain data from SLUTty at all, but we have now added this. And as more taxonomies are created, they will also be added to this tool. And more descriptions on what those taxonomies are can be viewed here. And if you have more information on specific algorithms, you'd like to look at the actual algorithm itself in GitHub. What were the marker genes? When should you use it and why? You know, recommended usage. We have that as well. We also have a number of R and Python notebooks associated with MapMyCells, which you can use here as well, just to get your data in the right file format, or if you have any questions, or you're interested in a use case. And again, like if you're interested in cross-species comparison, that you know, that is a potential use case for using MapMyCells. We do have to, there are some extra steps you have to do in converting in between species, but this potentially could be useful, you know, if you have mouse Alzheimer's data, transcriptomic data, and you'd like to compare it to the human Alzheimer's data that we have. This is one possible way to do that. It is optimized for, you know, for single species or working within a species. You know, this was not created to do cross species, but it is possible to try to do cross species if you'd like. And again, if you have any questions about it, post on our community forum. The multiple people involved with MapMyCells can help you troubleshoot, you know, any issues that you're having. Okay. And to actually go to MapMyCells, I have that. So I know we started late, so what time should I expect to be over? 12:15? You want to have it all at least? Is that fine? Yeah, that works for me. I have, yeah, I wasn't sure if I should spend more time on this or move on to the third thing. So I do have a third thing to mention. And it's kind of difficult to actually demo MapMyCells since it takes about to actually run it, the algorithm. It's not,
Let's move on.
it's like not.
The spirit, the main takeaway here is this mapping is a very important thing. Understand how your data, you might. So people want to do this in the way of preparing data sets, understanding something you have something new or how your data may relate to something. So the best way to do it is just to, it's really easy to use. Make an example. If you have the transcriptomic data of this nature, you can simply get on and try. So there's an R script which has data right there, and you can just run the example.
So, Rachel, could you talk a little bit more about mapping procedures that you use, either across individuals within the species or maybe across species as you just talking about? Do you do it principally through the same region identifying each individual or species? Or do you have a spatial three-dimensional coordinate? Right. You show it in the mouse. I'm not sure I've seen it in the human. And much it seems like it's a set of regions in biological sample, then analyzed, and you have a description of it. Do you have work? Mapping across the regions?
Yeah, so the healthy human data that we have from Slutty et al., yeah, that doesn't feature spatial transcriptomic data. That's just single nucleus data, and 105 different dissections were done across the 3 different brains for that. And I don't believe that has been mapped to the Allen Human Reference Atlas yet, but I think that's in the works.
It's a region-based, not global-based.
Right.
Yes.
It's not point based in the sense that MR or no, none of it is done. In the case of, say, that this, so we refer to this one app, Selenia. This is this proto standards in the lab. They just sampled, they went through it, and then after they had a first look at the distribution of transcriptomic structure of the brain, they went through and sampled about 100 different regions, but completely based on anatomical driven, not formative based in there. The way we compare between species is purely based on transcriptomic similarity. And the taxonomies we develop each of the species, and then there's an attempt to align these, showing that, oh, this is evident in this cell. This appears to be important on this cell. That's species. It's not done anyway. Yeah.
I was thinking, are we talking about the hitchhiker's guide to the mapping between ethnic species? Can you follow space and time evolution from mouse
Yeah.
to machines of human?
I really wouldn't be able to navigate that in all those dimensions, all along any of those paths, but the way we do it is by the way all this is done is the taxonomies. And the taxonomies are generated in each species, and then that which is enough common structure. We see the same between species, we get a reduced consensus.
So it's region-based.
Yeah.
Okay, you were talking a little earlier about generative AI being explored as a way to be able to create easier access. Do we envisage a time where we talk to our computer and say, Alan, can you show me everything you have on this positive neurons that support the idea?
Yeah, that's exactly the type of use case that, some of our data technology team is working on, right? Instead of having to go through and manually click all those different filters, can you just type in, show me statin cells?
Right.
Yeah, that code expressed layer 2 through yeah.
Like,
Yeah.
we are trying to build, we have a group that's trying to do that now. Also, we can more of an intelligent interface to the data, what can we do,
Yeah, because that's, the more time you spend with the atlas, the more filters you realize that are there, and the more possibilities you realize, oh, I can do this, this, and this. But it takes a little bit of time to sort of piece together how you would use all the, filter by anatomical area, filter by subject, human subject, dementia status, ApoE status. But if you could just type in, I want to know what this cell type and this cortical layer and this ApoE status looks like, and then get that would be much faster.
Like many data, I couldn't say like all, but like many data projects that in the databases that grow over years, they tend to grow somewhat osmotically, and then the organization of them is sometimes, you like it's evolution. You wouldn't necessarily build a human the way we have if you had a choice, and actually what would be the best way to do it every time.
It's called intelligent design.
Right, I think it should be a little bit more early intelligent design.
Yeah, and then the last tool that I'll be mentioning is the Cell Type Knowledge Explorer, which you were also briefly introduced to this morning. The Cell Type Knowledge Explorer is, again, a browser-based tool which features features some of our cross-species data. We have marmoset, human, and mouse. This is all primary motor cortex, and a consensus taxonomy was created from that transcriptomic data from the 3 different species where we can see cell type homologies. And again, it was the taxonomies were created using transcriptomic data, but practically the experiments were also done on the mouse data and also some of the human data as well. So we do have some of that morphology and electrophysiological information to help characterize those cell types. And this was all done in the primary motor cortex. So you saw this slide, some of these slides earlier this morning. This was a huge project, that mini atlas, that's not so many. It was 17 different studies were published. This involved hundreds of people across over 45 institutions. And, over 3 million cells were profiled from these 3 different brain areas, from these 3 different species. And what were some of the highlights? So here is the taxonomy, the transcriptomic taxonomy from the mouse. And again, since PatchPeak was done, these are physiological firing patterns that are labeled down here below the taxonomy, so we can see which firing patterns correspond to which transcriptomic types. And then we also, using PatchPeak, you're able to fill the cells from the inside of biocidin and then trace their morphologies. So we can see the morphological linkages between these cells as well. And this is all done from the mouse data. Here at the top, we have our human dendrogram here, which is showing that human primary motor cortex taxonomy. Then in the middle, we have marmoset, and then at the bottom, we have mouse. And these were combined to create a consensus taxonomy here. And on the right-hand side here, what we see is, which cell type are conserved across our 3 different species and which are not. Which cell types, like this LAMP5 cell all the way on the left, this inhibitory cell, there's a 1-to-1 ratio between our 3 different species. That's pretty well conserved across our different samples here. Whereas some of these other types, they're much more prevalent in one species versus another. And up here at the top, this dendrogram is actually color-coded to show the confidence mapping. So darker colored here means higher confidence, whereas this lighter colored red means lower confidence. But this is our consensus taxonomy from these 3 species within the primary motor cortex. And again, why primary motor cortex? Well, we know that, visual cortex is highly specialized in humans and non-human primate. Somatosensory cortex in mouse is very different from somatosensory cortex in human, but primary motor cortex is more similar across different species. And so we have a tool to access all of this data. So the Cell Type Knowledge Explorer is the name of the tool, and here's a QR code or a link, or you can also just type it in. Usually I can just type it in Google and it pops up. There's not really something else that has the same name. And here we have our 3 different species that I mentioned. So we have our marmoset, our human, and our mouse down here. And if you click on any of them, that is an entrance to that particular species data set. And so what's shown here is a sunburst plot. And so this is another way to show this taxonomy here, as opposed to a dendrogram. So this inner ring here shows our different classes of cells. We have our GABAergic here in red, our glutamatergic are blue, our non-neuronal cells are in gray, and our non-neural cells are greenish gray color. And then the next ring B shows us, the classes, the subclasses within the class. So down in GABAergic cells, we have our albumin cells, our statin, our VIP, our LAMP5. And then below that, in the outer ring, ring C, we have the different cell types that were defined. And these cell types, again, this was all based on purely just the transcriptomic information. This characterization did not include the electrophysiology or morphology data. And this plot is interactive, so if you click on any portion of this plot, it'll lead you to that particular. So I clicked on the GABAergic section, look at the GABAergic cells here. And what you'll see some plots showing the transcriptomic data. So we have a UMAP, we also have another plot showing the different gene expressions and the different marker genes that were used to determine that class. And then within each class, again, we have our subclasses. So you can click on any of these different subclasses by clicking on this drop arrow. At the subclass level, you get more information based on the morphology, the electrophysiology, and also some spatial transcriptomic data as well, and different marker genes. This is all from published data as well from that paper package from 2021. So we also have links to the papers themselves, and the method sections of these papers are extremely detailed. So I definitely recommend looking at them for specific questions. And we also have a little summary up here that just sort of, includes that summarizes all the data that's listed on the page. And then, the next level below our subclasses, we also have these cell types. Right now I'm in the LAMP 5 subclass, and if I click this drop down arrow, I actually get 8 cell types within that LAMP 5 subclass. And you can click on any of these and learn more information. And because this is a cross-species taxonomy, this is really the most exciting part, I think. So once you get down to the cell type levels, right now I'm at a specific cell type in the mouse. If I scroll all the way down to the bottom of the page, it shows me this heat map where it shows me cross-species comparisons. So what is the equivalent homologous cell type in the marmoset data or in the human data here? And on this heat map, the darker colors represent the homologous cell types here. And this heat map is also clickable, so you can click on the different parts, and then you can see, if I'm studying a particular cell type in mouse, does it exist in human? If it does, you can then click on that part of the heat map, and it'll lead you to the human version of the page and give you some of that information. Some use cases. Maybe you do mouse work and you want to know about their human equivalents. Again, this is only primary motor cortex data. So if you're, studying a subcortical region, cerebellum, wouldn't be super useful. Cortical areas, are more similar to each other. So maybe you're studying, visual cortex or somatosensory cortex, and you want to compare, something in primary motor cortex. You can use this tool for that. Again, this is all from healthy, non-diseased samples. So maybe you're studying a disease that affects the primary motor cortex, and you're doing, morphological analysis or transcriptomic analysis, and you want to compare your data to a healthy standard. That's one option. If you're interested in looking at, firing patterns and morphologies of genetically defined cell types, because we have hatch-seq data, that is a question you can explore with this data set. And also looking at layer-specific locations. So we have our morphology data, which can show you, is this particular cell type in a particular cortical region of a layer? Is it an upper layer 2-3 versus lower layer 2-3, right? That might be useful information for you in your experiments. And so if I go and actually go to the Cell Type Knowledge Explorer, I can just quickly click through things. So again, here is that consensus taxonomy here. We click down, to the mouse data, we get that big plot. We click on the glutamatergic section here. That leads me to this page showing me all of the transcriptomic information on the glutamatergic cell types. If I go over here to this arrow, that's where I get my different subclasses. So if I'm interested in my, layer 2-3 IT cells, I can click on that here. Get more information on the transcriptomic data, which marker genes were used to define this subclass. Morphologies, electrophysiological characteristics, that information is all on here. We also have some epigenetic data as well, because a tax-seq was done with these experiments as well. So we also have information on that. And the last layer of this taxonomy is the cell type. So down here, there were 3 different cell types that were identified within the layer 2-3 IT class. You can work on one of those. Read, which specific marker genes, when I'm looking at my 3 different cell types, which marker genes were used to differentiate between these 3 different cell types. And then the homologies here. Okay, so there were a lot of homologous cells in marmoset. The cell type, because all these are dark red. In human on here on the left, this one's not very homologous because it's a lighter color, but these up here were found to be homologous. And then if you click on any of these here, so this highest one here is 94% similarity. I'll click on that. And now it's leading me to the human page where I have information on this human cell type from the primary marker text, which marker genes were used to identify it here, and cross-species information as well. And a few more things I just wanted to highlight again before this ends. So again, we have our community forums. I mentioned this many times. It's super important. If you go to community.brain-map.org, this is our community forum. You can search different questions in different topics. We answer questions about anything related to our brain data here, not just the tools I showed, but pretty much anything. You know, people ask all sorts of questions. And most are answered by scientists who directly work on these projects and can help out with all their nitty-gritty questions and details. We also have this cell types resource catalog that me and one of my colleagues created just to help organize all of the cell type data here. So we have a number of links to useful things here. You know, if you want to learn more about how CatchSeq works, if you're more interested in, cell type nomenclature, if you're interested in, understanding your GitHub better, some of our protocols, and, supplemental data. We also have, use cases, we have video tutorials, written tutorials for using some of these tools. I mentioned acronym lists because not everyone uses the same acronyms when talking about different brain areas in mouse and in humans. So we have these acronym lists, which are just CSV files that are useful for understanding, some of the cell type names that we have and why a cell type has a given name. And annotation tables. These annotation tables are also very useful for understanding the cell types that are presented in our data sets. Why is that, what were the marker genes used? What area of the brain is it in? These annotation tables contain a lot of information. And just more information about these different data sets I talked about. That is also located on this page. And also, here's my email address. It's rachel.hostetler@alleninstitute.org. So feel free to email me if you have any questions. If you want to dive into any of these things I've talked about, please feel free to reach out to me. And thank you all for attending this talk. And the Allen Institute is a very large institute. There's over 900 employees right now. So a lot of people went into making these data sets. These are massive data sets. It's not just one person doing all this work. It's these very large teams. And so it's a huge collaborative effort to get this data analyzed and out there for others to use. All right. And I'll take, any more questions? Correct.
Yeah, so we'll get right into it. If we can try to learn about at an intersection, I think with the, BigBrain or all you can, I think it'd be nice. I think this gives you a flavor, kind of data and stuff. I think it's going to be